Hi, everybody, and welcome to Chapter 2 for the second week of Psychopharmacology at Northern Kentucky University. I am going to do a mini lecture about the brain, and this is just to set the context for all the work that we're going to do in the future chapters, looking at the effects of different psychoactive drugs. So some of this may be a bit of review for some of you. You may see this material and say, oh, I've seen this many times before. Some of you, it's a bit new. So I just want to make sure that everyone is at the same point. Okay, so the first thing that's really important is that psychoactive drugs alter brain activity. And so that's why psychoactive drugs affect emotions, behavior, cognition. The brain is the basis for all those sorts of things. And so if the psychoactive drug is affecting the brain, then you would see changes in emotions, behavior, cognition. Drugs, especially if they are used over a very long period of time, they can actually change the brain. And so that's why we have to think about when people are using psychoactive drugs, if this is a short-term use or long duration use, because that may change how the brain responds. The first thing that we need to do here is understand how the normal brain functions, and then we can sort of add in this extra layer of how the psychoactive drugs are affecting the brain. Okay, so the first thing we need to talk about is those cells in the brain, the neurons. They are the cells that are receiving and transmitting electrochemical information. Everything that we think about when we think about how we process emotional information, how we make decisions, how we do anything, that comes from this activity at the level of the neuron. And so neurons are central to understanding what's going on with psychoactive drugs, particularly the communication that the neurons use. So there are different parts of the neuron, and you probably recognize this. All of you have seen this in Introduction to Psychology. There's the cell body of the neuron. There's the dendrites, which are the receiving parts of the neuron, and the axons that send information away from the neuron and transmit it to the next neuron. And you can see on the picture that the dendrites look like trees, and you can remember axon and away, they both start with the letter A. That's an easy way to remember that axon is sending information away from the cell body. And then myelin is wrapping around the axon, and that speeds up transmission. Okay, so a little bit more about each of these parts. The cell body is that command center of the cell. It contains the nucleus, and the nucleus houses the genetic material. Okay, so we want to pay attention to that. The dendrites are the branch-like structures. The interesting thing about dendrites is they can change. So one of the things that is just sort of remarkable about dendrites is that if you are engaged in learning new information, the dendrites might grow. And if you're not engaging in information and you're very sedentary, the dendrites might shrink. So this is not just a static thing. These cells are changing based on experiences and also based on other things going on in the environment around the individual. They are the part that contains the receptor sites that receives information from other neurons. We'll get to that in a minute. So they are critically important if you add a drug into the system. The axon is the long rope-like extension that comes out of the cell body. As I said before, it starts with the letter A, so you can remember away from the cell body axon, and that helps send a message on to the next neuron. And it can be much, much longer than the diameter of the cell body, so it can run very long distances. So even in this picture, it looks long, but it could be much, much longer than that in reality. Remember, you would have to have a microscope to see these cells. They're so tiny. And myelin is the fatty tissue that covers the axons that helps insulate and protect the electrical current, allowing electrical messages to travel more rapidly. So at the very last chapter of this course, we'll talk about, different psychological disorders that come with aging, where myelin is declining. And so as a result, the efficiency of electrical communication declines. So it is important to just, have a bit of idea about that. And so when you look at the brain, so this is a picture of a human brain after autopsy. And the brain is cut in half. So right down the middle, like through the nose of the brain. Okay, so if you look at one half of the brain, if I turn the correct way on the screen, the back end here, where it looks like a little like cauliflower or something like that, that's the cerebellum. That's at the base of your brain. Okay, right at the base. So you can kind of have a sense that what you see on autopsy is something that looks lighter in color and something that looks darker in color. And so the lighter parts of the brain, the interior sections that look more white in color, those are the myelinated axons. And so that's where it gives the white appearance. The gray matter on the outside, so on the cortex that you would see on the exterior, that's more gray. And those don't have myelination. Okay, so when we talk about sending messages, that is what we call neurotransmission. And so when cells are communicating with each other, they do it to two types of signals. There's the electrical part that occurs within the neuron, and then there's the chemical part that occurs between neurons. And I'll show you a picture in a second so you'll see a little bit more clearly what I mean. It's the chemical part that's super important for psychoactive drugs, because when you add a drug into the system, the chemicals that normally get released between two neurons, you can add in something extra like a drug and it'll affect how those chemicals are working in the system. So it's the chemical piece that is really important that we will focus on. So it's not that the electrical signals, the action potentials that occur aren't important, they are, but in this course, when we talk about psychoactive drugs, we're talking about the chemical signals. And that's because a lot of the chemicals, the psychoactive drugs that people will use, look a lot like the naturally occurring chemicals that you have in your brain. And those naturally occurring chemicals are called neurotransmitters. Okay, so here's a picture now that shows the end of the first neuron and the end of the second neuron, and there is a little space. So you see these little dots everywhere. Those little dots are in a space called the synapse, and that is the space where the neurons actually are not touching. So you would have to zoom in really, really close, and you would see that they are not actually touching. There's a little space, and that's where the neurons are communicating using chemicals. And the neurotransmitters are the little chemicals that get released into this space. Now, if you ingest a drug from outside your body, you could also have chemicals from that drug in that space. Okay, so you can probably already see that that is the place where we really will see psychoactive drug action. Okay, so in the presynaptic or first neuron, when you're looking at sending a message, in the first neuron, you have an action potential, the electrical part that reaches the axon terminal, and then the vesicles containing that naturally occurring neurotransmitter, chemical that our brains make normally, they are triggered toward the synapse, and then those chemicals, those neurotransmitters, are released into the synapse, and they diffuse to the next postsynaptic neuron. Okay, and then the neurotransmitters are going to float by diffusion. So diffusion, just like imagine you have a cup of coffee and you put a teaspoon of sugar in your cup of coffee. If you put your teaspoon of sugar in there initially, it's going to be all the sugar in one spot. But if you leave it long enough, by diffusion, sugar will be everywhere in the cup of coffee. And that's kind of what happens. All the chemical is released on the first presynaptic neuron side. And then there's lots of chemical on that side and no chemical on the postsynaptic or second neuron side. So because of diffusion, it'll move towards where there's empty space, and then eventually it'll bump into the second neuron. And the neurotransmitters are going to attach to very specific receptors contained on the dendrites in the cell body of the second postsynaptic neuron. And that's when the electrical signal will be activated, and you will have continual electrical activity until the next neuron releases chemicals in the chemical part. So that's the activity. Okay, so I mentioned that there's these receptors. These are places where those chemicals can bind, and they bind like a key would bind into a lock. In other words, a certain neurotransmitter fits into a certain receptor. Okay, so you have to have the right chemical neurotransmitter and the right receptor. And if it bumps into it and fits into that place, then you'll have an action potential and you'll have an electrical signal. And sometimes those receptors are types of receptors, we'll talk about this in a second, that when it binds, it actually doesn't allow that next receptor to fire. So there's two possibilities. But the thing that is really important here is that if you think about keys and locks, sometimes you can put something into a lock. You can take a key and put it into a lock, and it doesn't have to be exactly the perfect key, and it might still turn the lock, right? So the same thing's true with psychoactive drugs. As long as it looks close enough to a naturally occurring neurotransmitter, it might do the same thing as your natural chemical. Okay, so that sort of gives you an idea about where the psychoactive drugs one of the ways where they might have action in the brain. They are mimicking what might be occurring with a natural chemical. Okay, so as I mentioned before, if the key that fits into the lock lets that next neuron have an action potential, that is what we would call an excitatory receptor. That's where the action potential is likely to happen. And things like dopamine receptors, serotonin receptors, I'll talk about different neurotransmitters in a second, but there are different types of neurotransmitters where this is likely to happen. Then there are also the kinds of receptors that are called inhibitory. Those are the kinds where you put the key into the lock, the key will go into the lock, but the lock won't turn, so the door won't open. And so those inhibitory neurotransmitters are the ones where the action potential is not likely to happen. GABA, adenosine, those are the examples. And I'll talk about a little bit more in a second about those. Okay, so you have all this happening. What happens after the receptor has, there's been this binding of the neurotransmitter with the receptor? Well, you can have a recycling process like reuptake, where the neurotransmitter is taken back up to the first neuron to be used again. So recycling is reuptake. It could also be that there are enzymes that deactivate the neurotransmitter after it's done its job, and enzyme breakdown can, break down that neurotransmitter, and that gets flushed out of the system, urine or whatever. And so, different ways to use, either reuse the chemical or get rid of the chemical after it's done its job. Okay, so neurotransmitters, there are actually 50, more than 50 neurotransmitters, and probably more that we don't even know about. They're just under intense investigation. I know some of you are neuroscience majors, and you go into greater detail in a lot of different courses about this, and probably even do research on this in your labs. Neurotransmitters can have different subtypes, and so we're just going to talk about or focus on the ones that matter for the major drugs of abuse. And over time, even over my career, that has changed actually considerably about understanding what neurotransmitters are involved in which certain drugs of abuse. So, when I've done work over the years, initially, it wasn't really clear that adenosine had a lot to do with the effects of alcohol. They thought it was just maybe something to do with the effects of caffeine on the brain. And then over time, more recently, there's been more work. We realized that, adenosine is really involved in the effects of alcohol on someone's brain. So sort of shifting landscape. Now, in your textbook, there's sort of a table 2.1 that kind of gives you a snapshot of the major neurotransmitters we're going to focus on. So this is a good place to just take a look and sort of get an overview. If you are a little overwhelmed by the information in this chapter, don't worry, because in each subsequent chapter, we'll focus on one psychoactive drug. And so there may be only one or two neurotransmitters that you would need to know about for that chapter. And so as a result, what I really want you to get out of this chapter is more just like a big picture overview of, you know, what are the major neurotransmitters, just, you know, there are several involved, and I'll give you a couple highlights for a moment here of things to just take away from this chapter. Okay, so the first thing is dopamine will come up over and over again as a neurotransmitter. As a chemical in our brain, it's involved in so many things. The regulation of movement, our emotions, thinking or cognition, our motivation to do things and achieve things, and our feelings of pleasure. Because of its role in pleasure, that's why dopamine plays such a considerable role in drugs of abuse. Drugs are used because they're pleasurable. We use them for other reasons, like I mentioned in chapter 1, you know, maybe to optimize performance or to take away pain, things like that. But, you know, initially, a lot of times why people are using drugs is for pleasure. And so dopamine plays a considerable role in that. And it's not something that is just inconsequential. It's an incredible change that occurs with dopamine when someone uses a drug of abuse. So when someone uses morphine or heroin or any of the drugs of abuse, the amount of dopamine that's released is 2 to 10 times what our brain would normally release for natural rewards. So if there are certain things that we do that are natural rewards, you eat foods and you feel pleasure. Your brain rewards you for eating because you need to eat. Your brain will reward you for sexual activity because your body wants to reproduce. So you will have dopamine release for sexual activity, for eating, for lots of different things. But the amount of dopamine that's released is never what it is from an exogenous drug of abuse, like heroin, for example. And so this is the real problem, is that external drugs of abuse are hijacking the brain's normal activity and making a reward so much more pronounced than we could ever have naturally. And so for people who become addicted to certain substances, it's really hard for them to go back to that baseline state because they're so used to exceedingly amounts of pleasure from large amounts of dopamine release that getting back to a normal level is really difficult. The other thing is when you spend a lot of time releasing tons and tons of dopamine from a drug being in the system, if you suddenly take that drug away, your brain has stopped making dopamine at that same level. And so it's actually deficient. Okay. And so we'll talk about that some more in later, but it's an important thing to know that, you know, when you muck with brain chemistry, there are repercussions. And so at least from this chapter, I want you to take away that, okay, we have a certain kind of baseline level of things that we're doing, and then we're going to add in something from the outside. What does that do to the brain? Where does that, you know, mess things up with the normal balance? Okay. Norepinephrine is another good one to know about. It's important for emotional arousal, regulation of hunger and alertness. It is important for the fight or flight response. And some of the stimulant drugs, if you put them in the system, they'll elicit a flight or fight response, you know, elevation of heart rate, blood pressure, pupils get larger, you know, those sorts of things that help you react quickly to emergency situations. An external drug can set you in that state. And also when we get to the later chapters of the course, when we talk about psychological disorders like major depression and attention deficit hyperactivity disorder, sometimes when we look at individuals who are diagnosed with those disorders, they don't have enough norepinephrine. It's insufficient. And so that's another consideration, is that in disorders, sometimes you don't have enough activity of a certain chemical, and you might have to boost that activity of that particular chemical with medication so the person gets to more of a baseline state that you would expect. Serotonin is another neurotransmitter. It's really important for sleep, mood, appetite, sexual motivation, body temperature even. And again, just like I mentioned in the previous slide, in various psychiatric disorders, for example, someone with the diagnosis of major depression, they might have insufficient levels of serotonin. And so they may have a lot of trouble having normal sleep patterns. And once that serotonin level is reestablished, maybe with medications like antidepressant therapy, their sleep gets a lot better. And then they feel better. So sleep and mood are very tightly tied. If you don't sleep well, you kind of feel low mood. And when the sleep is restored, the mood tends to improve. Also, when we talk about the hallucinogens, like LSD in one of the very later chapters, they can also stimulate serotonin activity. And so that leads to a very interesting area that is sort of new, where researchers are looking at, can we give people hallucinogens to help them recover from a psychological disorder? And so there's been some work on hallucinogenic drug therapy to treat disorders like post-traumatic stress disorder in veterans and things like that. So that'll be coming later in the course. Acetylcholine is the first neurotransmitter discovered, found in the brain and skeletal muscles, where the nerves are meeting muscle at the neuromuscular junction. Acetylcholine will be important when we talk about nicotine in the chapter on nicotine and smoking. And it's also really important for memory, attention, and sensory processing. So interestingly, nicotine it is really important as a potential therapy if cognition is impaired in some disorders. It can also make you really nauseous. So what researchers are trying to figure out is, can we enhance acetylcholine activity by nicotine or nicotine type compounds to help people with various degenerative dementia disorders like Alzheimer's disease or something like that. Endorphins are large molecules called peptides, and there are natural painkillers. So if you, get in some sort of like accident, like a bear is chasing you and the bear bites you, it's not really great to curl up in a little ball in that moment. You actually should probably run away from the bear so that the bear doesn't finish you off, right? So, we have this natural painkiller to help us in these sort of emergency situations so we could survive. And so it's great. These endorphins release, the release of endorphins will help us block pain and make us feel good. Again, though, psychoactive drugs can hijack that mechanism, right? And so blocking pain, feeling good, that's something that, a drug can overdo in a person. GABA is another abundant neurotransmitter. A lot of research right now on GABA in various forms. If you want to talk to somebody who's really interested in GABA in our department, you can talk to Dr. Justin Yates. He does a lot of work on GABA in his lab. It's a really important inhibitory neurotransmitter, causes feelings of relaxation and sedation. And so a lot of the anti-anxiety medications act on GABA. GABA is also involved in the effects of alcohol or any of the other sedative drugs. So we'll come back to that later in the course as well. Glutamate, another abundant excitatory neurotransmitter, really important in learning and memory. And the changes in synapses, the synaptic plasticity. So we'll come back to that in the course as well. And then adenosine, which is my favorite neurotransmitter. I think it's understudied. It's an inhibitory neurotransmitter. It's found in both central and peripheral nervous system. And the reason why it's my favorite neurotransmitter is because my career, I spent a lot of time in my career looking at the effects of caffeine and alcohol in combination. So I did a lot of research on energy drinks and alcohol, looking at the combination of a stimulant and a sedative at the same time, and why would people combine those two substances. And adenosine is so central to that area, because we know that adenosine, those levels naturally rise over the course of the day and are believed to promote sleep. Caffeine comes in and blocks the action of adenosine, and that's why it increases wakefulness. So it's very interesting. Caffeine really doesn't do very much if you're wide awake, but if you get really sleepy, that's when caffeine really has a big effect, and that's why adenosine plays a role here. And then also, when you consume alcohol, you increase the levels of adenosine. That's why sometimes you feel sleepy if you drink alcohol. So if you add in caffeine, you would be blocking some of that sedation. And so that's why people might drink longer. They may drink more than they normally would intend, because normally sleepiness is a good protective mechanism that tells you, you should stop drinking, you're done for the night. That cue, that sedation cue gets missing if you add in something that influences the adenosine action, and caffeine is a good example of that. Any other stimulant might also do the same thing. So my favorite neurotransmitter, I'll be on the quiz. Anandamide. Anandamide is really interesting because it's more of a recent finding, discovered in 1992. And as you can tell, neuroscience is just, a field that continues to grow at leaps and bounds. And anandamide is really interesting because it's the neurotransmitter that really has the action on the cannabinoid receptors that is involved in the action of marijuana, or THC in marijuana. And so when we talk about the marijuana chapter, we're going to talk a lot about anandamide. It's really involved in things like appetite and sleep and pain. So that's also why people are studying it, because they want to see, if we understand anandamide, could we develop new medications that help people with pain? Because a lot of the pain medications that are currently used don't rely on the action of anandamide, and they're very addictive. But if you look at anandamide, people who use marijuana to treat pain tend not to be addicted to the same degree as other classic pain medications. So this is a very hot area of research, continues to be very interesting, and lots of new things almost every day. Okay, so just to recap here, the actions of neurotransmitters, they can increase activity via excitation or decrease activity in the brain via inhibition. Agonists are any drug that binds to receptors and acts like that neurotransmitter or mimics it. It fits into the lock. The key fits directly into the lock and turns the lock, just like the normal key would. And so, for example, heroin, it will bind to and activate the endorphin receptor. An antagonist is one that binds to the receptor, but blocks the neurotransmitter from binding. Okay, so it prevents that neuron from firing. So naloxone would be a good example, where it will bind to the endorphin receptor, but doesn't activate it. It blocks it, does the opposite effect. So if someone has taken a whole bunch of heroin and their breathing is impacted, and you quickly need to do something about that overdose, you would want to give naloxone, because it would block the effects and do the opposite. Okay. psychoactive drugs can do all kinds of things. They can increase the new synthesization of neurotransmitters. It can affect the transport or how neurotransmitters are stored, released, broken down. When we come to a specific example, I'll be explaining this in detail, but for the most part, you have naturally occurring chemicals, and then, you add in another chemical from outside the body, a psychoactive drug, and you're going to probably influence the balance and actions of the naturally occurring system of chemicals. Okay, so we were way down at the cell level of the neurons. Now, big picture about the nervous system, just a little bit of overview. You have your central nervous system, the brain and spinal cord, and then you have your peripheral nervous system, which is basically everything that's not brain and spinal cord, right? And so when you look at the peripheral nervous system, there's parts of the peripheral nervous system that are the sensory and motor nerves. So if I wave my hand, I'm relying on, activity of the motor nerves to do that. There's also part of the peripheral nervous system that you don't have any control over. Those are those internal systems. That's called autonomic, and it splits down into the sympathetic side and the parasympathetic side. Now, I just want to briefly explain this because drugs do have an impact on this. So the sympathetic side of that system is what comes into play when the bear bites your arm. You're out camping, suddenly there's a bear, the bear wants your food, so the bear bites you. You're totally in this fight or flight moment of, what am I going to do? Am I going to run away as fast as I can? Sounds like a good idea. So when that part of the system is activated, it uses lots of energy. Your pupils are open, your pulse is quickened, you're going to have sweating, you're going to do everything that you need to do to get out of that emergency situation. And of course, there are certain things you don't need to do in that moment. So your stomach is less active. And so your stomach you may have indigestion issues because you're not really digesting food properly at that moment. So, but that's okay. You're running away from a bear. But when this system gets activated because you have a speech or something that you need to give in class, or you're really, panicked because, you're on a first date and things aren't going well, then you might have upset stomach troubles. That's that sympathetic side. We don't use that very often, or we shouldn't use it too often because it really it's hard on the body. The parasympathetic side is where you conserve energy. And so you have normal digestion, you don't have this heightened panic response. And so what happens sometimes with the stimulant drugs is they really activate the sympathetic side. So if someone takes a lot of cocaine, they may be sweating a lot. They may look like they are, in a race. They may have faster pulse. And if they use cocaine a lot, that's really hard on the cardiovascular system. And so they may actually be prone to heart attacks and things like that because their body is spending far too much time in this sympathetic side. Okay, so big picture about the brain. The brain is, that complex organ. And there are three parts of the brain. There's the lower level sections of the brain closest to the spinal cord called the hindbrain. The midbrain is the tiny middle section, and the forebrain is the whole top part of the brain. And you have an organ that is, it doesn't look it, but it's just incredibly complex, filled with billions of neurons. It may only be 3 pounds, but boy, does it do amazing things, right? Okay, so at least for the lower levels parts of the brain, the medulla is the one part of the brain that I do want to mention because it controls breathing and heart rate. And some drugs of abuse, they can slow breathing and heart rate to the point that if you ingest so much drug, you can die from an overdose. So heroin, alcohol, any of the sedative drugs, those actions of way too much of that drug on the medulla is why we have overdose deaths. And so that is a part of the brain you really you want to be cautious of. And then drugs like marijuana, the one good thing about marijuana is that it does not affect breathing and heart rate. So even though it may affect other things, cognitive functioning, things like that, it would not affect breathing and heart rate. So you couldn't actually have someone die from too much of that. Okay, now, the part of the brain that will come up again and again and again in the course is the prefrontal cortex. That's the part of your brain near your forehead, the front part of your brain. And the reason why it comes up over and over again is this is the part of the brain that is the last to develop. So brain development, the parts of the low part of the brain, those look really similar between us and a frog. Really not much different. But the top part of the brain, this is what really makes us human. It's the complex decision making, the thinking about your future, all that part of your brain. It takes a long time to develop. In some studies, it's ages 21 to 24 before we really see full development of that section of the brain. And so that's the part of the brain that tells you to maybe consider to stop doing something. And individuals who have a lot of challenges with substance abuse usually start using substances when they're in their adolescence. And that's when that part of the brain isn't really working very well. So we will come back to this part of the brain over and over again because it's so central to using drugs. The other thing that is so central, if you remember earlier, I just mentioned that we have dopamine. Dopamine makes you feel that tremendous amount of reward and pleasure. That mesolimbic dopamine pathway that we discovered, I didn't discover, it was discovered in the 1950s. It runs in the center part of your brain from the nucleus accumbens to the ventral tegmental area of the frontal cortex. And it's so rich with dopamine so that if you have an electrode in that anywhere in that pathway. So say you have a rat in the lab and you put in an electrode and you allow the rat to stimulate that area of the brain, the rat will just like sit there all day pressing a lever to get dopamine released in this pathway. It's the pleasure center of the brain. And the pleasure center of the brain is where all that release of dopamine occurs. And most drugs of abuse cause release of dopamine in this pathway. So you will see this also over and over again. And a lot of times those two areas are considered like the go and the stop system of your brain. So the mesolimbic reward pathway is like the go system of your brain that motivates you to do certain things, engage in actions that result in pleasure. The prefrontal cortex is the part of your brain that says, whoa, stop. Is this actually what I want to do right now? Is this the right thing to do? That weighs in higher order thinking to see how you're doing. And that's the end of this lecture. So let me know if you have any questions, and have a great week, everybody.